cell model.
Bacteria are prokaryotic organisms, which means they are single-celled organisms that do not have a true nucleus or membrane-bound organelles. Despite their simple structure, bacterial cells produce all the essential life processes needed for survival.
To create our cell model, we use recyclable and locally available materials.
Water bottle for capsule, electric wire for flagellum, toothpicks for pili and membrane, emerald green paint for the color of the cell, felt paper for the plasma membrane, cell wall, and cytoplasm, clay, old clay for ribosome, nucleus, and plasmid, cardboard box as the base of the capsule, brushes for us to paint the capsule, colored bond paper and colored markers for parts of the bacterial cell, stick glue, use glue gun to stick them all together, glue gun to stick all the materials together, cutter, cutting out the water bottle, 1/8 illustration board for the platform of the project, and lastly, popsicle for labeling the parts of the bacterial cell.
Before discussing the cell membrane, let us first identify the major parts of the bacterial cell and their functions.
This is the cell capsule. It is the outermost layer that surrounds some bacteria. It is usually made of sugar and acts like a coat. The capsule helps bacteria avoid being attacked by the immune system, prevents them from drying out, and helps them stick to surfaces. This is the cell wall, the rigid outer layer of a bacterial cell. Its main function is to protect the cell and maintain its shape. It also prevents the cell from bursting when water enters. The bacterial cell wall is mainly made of a substance called peptidoglycan.
This is a cell membrane, also known as the plasma membrane. It's a thin flexible layer that surrounds the bacterial cells and acts like a security gate. It is selectively permeable, meaning it regulates the movement of substance into and out of the cell. This helps maintain a stable internal environment necessary for the cell survival.
This is the cytoplasm. Cytoplasm is the gel-like substance inside the bacterial cells. It fuels the inside of the cells and holds the important parts of the cells, like ribosome, DNA, and plasmid. Since bacteria do not have membrane-bound organelles, most of the cell's activity, just as making proteins and breaking down nutrients of energy, happen in the cytoplasm. It also helps move nutrients and other materials around the cells, so the bacterium can function properly and survive.
This is the nucleus. The nucleus is the region that contains the bacterial cell's DNA. It stores the genetic information that controls the cell's growth, reproduction, and overall activities. Unlike plant and animal cells, bacteria do not have a membrane-bound nucleus, so their DNA remains concentrated within the nuclear region. These are the ribosomes. The ribosome serves as the protein-producing structure of the bacterial cell. Its primary function is to synthesize protein by translating the genetic instructions stored in the DNA. These proteins are essential for the cell's growth, repair, and other vital biological processes.
This is plasmid. Plasmid are small circular pieces of DNA that are found in bacteria. They are separated from the bacteria's main DNA and carry extra genes to help the bacteria survive, such as genes for antibiotic resistance.
These are pili and membrane. Both are a hair-like structure on the outside of bacteria. Pili are shorter, more numerous, and work like tiny hooks to help bacteria stick tight to surfaces or other cells. Pili are longer and fewer in number and help with sticking to. Plus, they let bacteria share genetic material or move around. Both help bacteria survive and connect to their surroundings.
Flagellum. This is the flagellum part of the bacterial cell. The flagellum is a microscopic like appendage that helps cells move. It acts like a tiny motor, allowing microorganisms swim through liquids and avoid harmful substances.
Although our model shows the bacterial cell as a whole rather than a magnified view, this pink layer represents the cell membrane. This structure is explained by the fluid mosaic model. The fluid mosaic model explains that the bacterial cell membrane is mainly composed of a phospholipid bilayer and embedded with membrane proteins. The phospholipid bilayer acts as a selective, permeable barrier, while membrane proteins help transport different substances across the membrane. It is called fluid because the substances can move within the membrane, and mosaic because of the different molecules working together.
The cell membrane, the pink layer of our model, is a selective permeable, which means it controls what enters and leaves in the cell to maintain the cellular homeostasis. Diffusion is allowing substances like oxygen to move into the cell and carbon dioxide to move out from an area from high concentration to low concentration without using energy. Osmosis is moving the water to the cell to maintain the cell water's balance. Active transport uses energy to move nutrients into the cell against the concentration gradient when they are needed. Together, this membrane transport mechanism helps the bacterial cell to maintain a stable external environment, allowing it to grow, function properly, and survive despite changes its surroundings.
To sum it all up, our model demonstrates how different parts of the bacterial cell work together to support life and carry out essential functions. The cell membrane helps regulate the movement of substances and helps maintain cellular homeostasis through the fluid mosaic model. This reminds us that although bacterial cells are microscopic, they possess remarkable structures and processes that enable them to thrive, survive, and adapt to different environments.
The oneness of